Hi guys, welcome to week 2 screencast. This week we are going to start talking about cell reproduction, so mitosis and meiosis. We're going to start actually with a quick review on chromosomes before we start talking about how they behave and what they do. So this is the video that I'll be asking you to watch to go along with this, just so you can see it. And now we can start our slideshow. So again, starting to talk about chromosomes, I think you probably all know a little bit about what chromosomes are, but we're going to get into a little bit of detail. So when we talk about how they behave during cell division or cell reproduction, it makes a little bit more sense. Fancy. Okay, so I do have a question for you guys. And I know you can't talk to your partner right now, but I want you to really pause this and take a moment to think about the question. Why do cells need to divide? There are 3 reasons. And what would happen if cells did not divide? So pause right here and try to jot something down. Okay, hopefully you took a pause and wrote that down. Cells need to divide to do these 3 things: to repair, right? So if you fall down and scrape your knee, that scrape doesn't stay there forever. That skin eventually knits itself back together, and maybe you have a scar, but it's all healed up. Cells divide to replace other cells because, just like everything, cells are not immortal and they'll need to be replaced. And thirdly, for growth, right? When you're born, you're maybe what, a 7 to 9 pound infant, and now you're a teenager. So all of that growth is essentially due to cell division. Now, when we talk about cell division, what we're thinking about are these structures inside the nucleus that contains all of the genetic information, right? This is where our genes live, and our genes are all of the instructions for everything that our bodies do, everything our bodies make, all of the traits that you see as you. Now, there's a couple of ways that we can think about chromosomes or genetic material, right? And I want you to look at this diagram. So when a cell is not dividing, when it's just doing whatever its daily job is, the genetic material is inside the nucleus in the form of chromatin. It's almost like a big bowl of spaghetti, ramen if you prefer. That material is all loosey-goosey, unwound, and all mashed together. When a cell is getting ready to divide, those materials need to get organized because in order for the cell to divide, you need a perfect split, right? Everything needs to be copied perfectly and everything needs to split in half perfectly. So what will happen is all this genetic material will tighten up, it will coil up, and it will wrap itself around proteins called histones until it's condensed into what are actually chromosomes, which are these X-shaped structures that I think, at least to me, are like the classic chromosome shape. Okay, now sorry, I'm going ahead a little bit. Let's talk a little bit about the structure of this chromosome. Okay, each chromosome, this X shape, has 2 identical halves. These halves are called chromatids. Now, as the cell is getting ready to divide, it needs to make copies of everything. So essentially, what you have are 2 exact copies of the same chromosome that are attached in the middle. That attachment point is called a centromere. It's almost like a belt. It like cinches the 2 parts together. That's important because eventually that is going to come apart and these 2 chromatids are going to split apart so that the cell can divide. One thing to note about a chromosome is that it has a structure called a telomere. I always compare this to the little piece of plastic on the end of a shoelace, right? An aglet. This doesn't code for any genes. It doesn't have any true genetic code or material. However, they protect the chromosomes, like an aglet protects your shoelace. It keeps it from unraveling. It keeps these arms from making inappropriate bonds, fusing into rings, or attaching to other pieces of DNA. Because, again, I'll say it a million times, when you think about cells dividing, you need two perfect copies so that each cell ends up with one perfect copy. Just a quick thing about prokaryotes, and they address this in the video you're going to watch. In general, a prokaryote will have one circular piece of DNA in order for it to reproduce. We don't need to go too much into that. Just a quick word about chromosome numbers, and we'll talk about what diploid means in a moment. The number of chromosomes does not necessarily relate to how complex something is, right? So here are humans down at 46 chromosomes, and there's tons of organisms that have more than us. Sheep at 54, horse at 64, dogs at 78. So chromosome number does not necessarily relate to complexity. Okay. One of the things that we definitely need to talk about, and we'll talk about this more during actual mitosis and meiosis, is the differences in types of chromosomes. So we have total 46 chromosomes or 23 pairs, right? If you've done 23andMe, that 23 relates to how many pairs of chromosomes you have. The first 22 pairs are what we call autosomes. These code for everything in your body. Skin color, hair color, traits, protein production, every single thing that your cells need to do or make. The 23rd pair are called the sex chromosomes. These are your X chromosome and your Y chromosome. This is a boy, X and Y. If you were looking at a female, this is called a karyotype. If you were looking at a female karyotype, you would have two copies of the X chromosome. So these behave a little bit differently. And as we get into our discussion, we'll talk a little bit more about that and a little bit more about how that impacts how traits are passed on, specifically how traits are passed on in males, because this does change some of the diseases, disorders, traits that a male can get from their parents. Now, when we talk about meiosis, we'll get more into how this all works. But essentially, this is the basics. Males have an X and a Y. Females have two X chromosomes. The father is who determines the gender or the sex of offspring. We'll talk more about that later. One of the things that I will be asking you to read, excuse me, is about homologous chromosomes. Now, since we all receive one chromosome from mom, one chromosome from dad, we've got two basically variations of chromosomes that code for the same thing. So let's say, I don't know, let's say this is hair color, right? You're going to have one of those alleles, we'll say, from mom and one from dad. But when your chromosomes are replicating in order to reproduce, you have to look at the similarities between. So these two here are what we call sister chromatids, meaning they are exactly alike. They are the exact same copy of a chromosome. However, one from mom, one from dad. This is what we call a homologous chromosome, meaning this is coding for the same thing, right? So I said this was hair color from mom. This would be your hair color gene from dad. They code for the same things, but they are from different parents. The ones that are attached are sisters, and then these two are homologs. One of the other things, that you'll be looking at is something called a karyotype. This is an arrangement of all the chromosomes in a cell, and this is generally used to look for abnormalities. During certain phases of the cell cycle, chromosomes can be captured basically in a photograph, and they can be aligned with their pair. You will be looking at an online karyotype to assess for abnormalities, but essentially, you should have two pairs of every every chromosome. X and Y may not be a pair in that they are not identical, but they will pair up as the sex chromosomes, and these should be alike. If you have an instance where you have an extra chromosome or you are missing a chromosome, you could also have an extra X or an extra Y. Those are all indicators of disorders, and that's something that a doctor or a genetic counselor would look at on a karyotype to assess. Now, this is, I think, the last thing we're going to talk about. Yes. So, the last thing we need to talk about in terms of chromosomes is haploid versus diploid. And this is going to be important when it's time for us to think about cells reproducing, specifically, or I should say, especially when it's time for us to think about the production of sex cells or gametes, which are your sperm cells and your egg cells. So, haploid. If you are referring to a cell as haploid, what that means is H for haploid, H for half. There is half the number of chromosomes in the diploid cell, meaning one copy of every homologous pair and one sex chromosome. So when you think about, let's say, a human egg cell, that cell is designed to combine with another cell. So these will each have half the required chromosomes so that when they come together to make the zygote, you end up with a diploid cell, which is the total number of chromosomes that a cell needs. So, diploid cell, meaning there are two autosomes for each homologous pair, meaning one copy from mom, one copy from dad in each pair. Our diploid number, humans, is 46, right? So we have total 46 chromosomes. That is from 23 pairs of chromosomes. If you are looking at gametes, which again are your egg cells and your sperm cells, those will have half of the required number of chromosomes so that when they combine, you get 46. So human diploid cells have 46 chromosomes. Human haploid cells, which are the sperm and the egg, have 23. And that is the end of our slideshow. I hope this was helpful. Please refer to the video that is posted as well for what are chromosomes and take a look at the karyotype activity, and we could come together with questions. Okay, thanks everyone.